This is a production of Cornell University.
So thank you for the invitation to come down and speak. It's a beautiful sunny day in Ithaca, so I'll try to talk fast so you can get back out there in the sunshine. So this is my title. I work for a company that develops biological seed treatments, and what I mean by that is fungi and bacteria that you can coat the seed with, plant it in the ground, and they affect plant performance. And the main fungal species that we're working with is Trichoderma. That's what our existing products are, and this Phytobiomes, microbiome approach to research is something that is really important in what we do, because if you think about it, what we are leveraging is the phytobiome in our technology. So this is a brief outline of my talk, and I realized looking at this that I'm only going to partially follow this. Things are a little out of order, but you'll figure it out. So before I jump right into things, there are a couple things I want to talk about to the students and postdocs in the room. And the first thing is careers. And so you all may think that you know exactly what you want to do, but it's really rare that you take that straight line path and you end up doing what you think you're going to do. So I never thought I would be the chief science officer at a company. And my main message is think creatively and broadly about what your career options are. And the other thing I would say is that dollars are rare, are few and far between, regardless of the sector of science that you're in. Academics, industry, government, anywhere you go, you're going to be operating on a shoestring, unless you're working in the plant breeding program in Monsanto, where you can afford to put out 500,000 plots every year. So just to keep that in mind. Okay, the next thing I want to mention is about communicating science. This is something that I'm very passionate about. I think that everyone here should think about, because you have a lot to communicate about. You have a lot to think about in your careers doing science, and one of them should be how you talk about your science. Okay, scientists until fairly recently used to be in a position of authority, that we were seen as experts in the field, and we worked with facts and things like this. And so there's been a change. Okay, so we need to think about how to speak about science and how to communicate that to the general public. So these are just some notes that I have. If you don't know how to speak about science, if you're not a good speaker, figure out how to do that, right? That's going to be really important. You need to be able to communicate science. Not everyone is going to argue like a scientist does, right? We work with facts, we have discourse, but that's not how everyone does it. And the other part about this is don't just talk to scientists, okay? We can publish in peer-reviewed journals, we can have really nice conversations at scientific meetings that we go to, but there are other people out there that you need to be talking to, and there are other non-scientists who are talking about science, right? The bloggers, for example, are going to go into that peer-reviewed literature as well and mine it for what they're interested in. So we need to learn how to talk to these people and figure out how to be part of the conversation. And so this is what I'm talking about. Just a note about, bloggers and things like this. People will question your motivations, right? That's the whole goal of some people when they question science is, why did you do this research and who's funding it, okay? So these are the things we need to be thinking about. You may rationalize why you're doing research on a scientific level, but other people are going to find other reasons. And so I just want to bring up these points as things we need to be thinking about as future scientists. Okay, the rest of this should be fairly obviously. Think creatively about how you make yourself heard. I don't know what the answer to this is. I'm not good at Twitter. I can't even figure out how to send a tweet. That's just my generation, I suppose. Listen to what people are saying and teach as often as you can. Okay, moving on. I work for a company called Advanced Biological Marketing. We are based in Van Wert, Ohio, and the company was founded in the year 2000. Our R&D lab is located in Geneva, pictured here, probably the day after we moved in. Notice how nice and clean it is. It looks nothing like that right now. So Gary Harmon, who was faculty at Cornell up in Geneva for 40+ years, was our first chief science officer. He has since retired. And Gary Bergstrom asked me, what does that mean? Well, he's starting another LLC. So, so anyway, this is where I work. And this is what advanced biological marketing does. Discovery, development, production, and sales of microbial seed treatments. These microbial seed treatments are generally referred to in the industry as biologicals. So when I refer to biologicals to a scientist, that means a lot of different things. In my industry, it means these living seed or other types of applications. We're working primarily with Trichoderma, but also Bacillus and Rhizobiome because we have products for lots of different legume crops. What we are selling are spores. And in the case of more fragile organisms like the gram-negatives, we're selling encapsulated cells. And this is an important thing because what we do is we sell a product that is put on the seed, might be combined with agricultural chemicals like fungicides, insecticides, things like this. And so what we're actually selling is an important thing to think about. And how these are used, well, they're used by the farmer all the way up to a commercial seed treater in a lot of different environments. And I put this picture in just to show some of the differences, and I'll show you, more than just this, but this was actually a field trial that was done this year in a drought situation where this, you can kind of see these strips, and this is typical of some of the kinds of data that we collect. So a strip trial where this very green strip was the control standard chemical treatment, and it was not tasseled yet, and these are a couple of biological treatments. Our treatment actually won this particular trial, and you can see some of the differences here in the different corn and the different cobs. These from the standard chemical treatment didn't, oh, I guess I have a pointer, sorry. So, not that you can see it, okay. So, on your left are cobs that didn't fill, they're slower maturing, as you can tell by the lighter color. And then those on the right were our product, which is called Sabrex, and that contains 2 different strains of Trichoderma. And it is matured at this point in the season, and it filled much better. Again, it didn't fill completely because I said this was under severe drought stress. So that's a little bit about the company and what we do. But there are a lot of other players in this game. So this is kind of different pedigrees of the companies that are also in seed, ag chemicals, and now biologicals. And this was fairly accurate until about a month ago because, maybe I'll use this as a pointer. So Bayer is now in the process of acquiring Monsanto. So if that goes through, that's a big deal. And Dow has, I think, finalized their merger with DuPont. So now, oh, and Syngenta is no longer just Syngenta. That's actually owned by a Chinese company called Kim China. So there are now actually just 4 players. BASF, in contrast, looks pretty small relative to these others. So so ABM plays in the same area, right? Where we're producing these biologicals, and all of these companies are as well. So we're a teeny tiny fish playing in a pond with some quite large fish. Okay, so what are biologicals? As I said, it's this general term. It started out as biological control, the use of one organism to limit the activity of another. And we think, when you look in the literature, most biological control is thinking about insects to control insects. But we're using fungi and bacteria as seed treatments, and we can control insects, nematodes, fungal and bacterial pathogens. There are several models about how this might work, and I'll get to that in just a moment. And this has obvious potential for agriculture, right? If you just think about disease control, well, that's one thing that obviously I'm speaking to the plant pathology department. We want to control disease, but there are a lot of other things that happen too. The thing that you can get with most any biological is growth promotion, right? You see plants grow bigger. They often look greener because the genes involved in photosynthesis are upregulated. We see increased yields, and so that benefit is fairly obvious. And so then if you can apply a safe biological product to the seed, or even as a foliar spray, or along with the fertilizer, maybe you can reduce the need for agricultural chemicals, which should be good, which in turn should lead to healthier and safer agricultural systems, more sustainable agricultural systems, right? So biologicals have been around, or biological control has been around for quite some time. The first registered fungal biocontrol was T22, and that was done by Gary Harmon in 1990 was when it was registered. It had clear demonstrations of this performance enhancement in the field and disease control in the field. And, but some of the people are, there are a lot of products that were developed at that time and since for biocontrol and as biologicals. And sometimes this whole industry was its own worst enemy because there wasn't a lot of data. It was poorly understood. People were brewing these things in their mom's basement and selling dirty water, right? So there's a lot that happened in the early days that is still biting us in the butt now, right? So hopefully I'll show you how we're moving beyond that. But there are several control mechanisms that were hypothesized at that time, and from your intro plant path course, hopefully you know some of these. Anabiosis, where an organism might produce a chemical or metabolite that is toxic or inhibitory to another organism. Niche replacement, where one organism outcompetes another, and a direct antagonism, which, did I show that on the previous slide? I don't know. Where an organism might actually kill another organism, not necessarily through an anabiosis method. And then performance enhancement mechanisms, there are several of those, and one of the main mechanisms that's hypothesized is nutrient mobilization or bioavailability. And also through growth promotion, you get with that increased root surface area, which has obvious benefits in terms of water and nutrient uptake. So this is kind of where we started, but the problem was the field performance has been highly variable. There's no question about that. And as I said, some of that came to do with how some of these products were developed and produced in the early days, and that's what everyone in this field is trying to overcome. So moving on, what we know now is that the mechanisms do include, for disease control, do include this niche replacement, anabiosis, and mycoparasitism, but also there have been a lot of studies from Dr. Harmon's lab, as well as many others around the world, showing that there are many genes that are actually turned on when the plant is colonized by some of these biological agents. And so we get an induced systemic resistance of many different flavors. Abiotic stress resistance, we see that the changes in plant gene expression due to this colonization have a really big effect on reactive oxygen cycling. That is, reactive oxygen species, as we know, are stress molecules, and when the roots are colonized by Trichoderma specifically, the genes in this cycling pathway are upregulated, and so the plant is better able to buffer stress, to protect itself against stress, and protect its proteins and cell structure. So growth promotion, biostimulation, we know that, yes, soil nutrient availability is important here, but we also see that the changes in plant gene expression upregulate carbon metabolism and photosynthesis. So what we're seeing here is the hypotheses from the early days weren't wrong, but there's more to it than that the organisms are actually directly affecting how the plant is expressing its own genes. Okay, so I just want to give a few examples just in pictures here of some of these, some of the effects. And so this is an experiment that we do in the greenhouse for disease control, just one of many different types, because obviously we can test for anabiosis on a plate or in the lab. But what we care most about is testing these things in the soil and better yet, in the field. Okay, so this is a greenhouse experiment. And what we've done here is we've infested the soil to an LD50. So that is with Rhizoctonia. And so if we plant 10 wheat seeds in there, we expect to kill 5 of them. Okay, that's the assay. And so we've done a good job of that right here. We've got the control, and there's the control. And all of the rest of these were treated with the standard chemical treatment. And what that standard chemical treatment is a mixture of fungicides and insecticides. And it just depends on who you buy it from as to exactly what those are. So here's a standard chemical treatment, and you see that it's got a fungicide. It actually has 3 fungicides in it, and it's controlling the Rhizoctonia better than the control. But when you throw Trichoderma in that mix, then you see either with the standard chemical treatment or Trichoderma alone, we see a really big improvement. In fact, we have 100% germination in the Trichoderma shown here. So that's one way we do this assay. And you might say, oh, but you put a fungicide on the seeds. How is Trichoderma, which is a fungus, able to grow? Well, Trichoderma, first of all, we put on spores, which are very resistant. And the other thing is Trichoderma is actually broadly resistant to fungicides, which is one of the key reasons why we use it. Okay, so that's a disease test. So looking at abiotic stress tolerance, these are some plants that have that we've either applied a water deficit to, and we did that just by determining what the 100% water holding capacity was on that pot, weighed it, and then water to 60% of that level. And that's our water stress. Salt stress is we're watering with a salt solution. And the plants on the left did not have Trichoderma on them, and the plants on the right do have Trichoderma. So these pictures, I know it's just one plant, but it's a demonstration of the kind of effect that you can see here. And as I said earlier, we want this to be able to translate to the field. And so we do a lot of work in the greenhouse, but as fast as we can, we move our pipeline products out to the field so we can get some real world data on them. And so this was an entire field that was split down the middle. Half was planted with no Trichoderma. The other half was planted with Trichoderma, which is, in this case, was our Sabrex for corn product carrying 2 Trichoderma strains. That treatment was made at the time of planting. And both of these were also coated with the standard chemical treatment, okay? So they received the same base treatment. The plants on the right received our Sabrex for corn, Trichoderma. This was at the end of the season. Illinois in 2012, there was a severe drought, and so you can see the result. The Trichoderma enabled the plants on the right to continue to live, to produce a yield, which you can see in the cobs below. It wasn't perfect ear filling down here, but it was far better than the untreated plants on the left. Okay, the plants on the left, we don't have yield data on or what the yield improvement was on this because the material on the left was not harvested. It was just too poor. So, okay, and then finally looking at growth promotion, as I said, this is the trait that is most commonly affected by a biological product. It may or may not control disease, it may or may not improve abiotic stress, but it will like 95% of the time give you a bigger plant. And so we can, we have a Sabrex product here for rice, and so this is showing standard chemical treatment alone on the left, and Sabrex for rice over that standard chemical control, and you can see the larger root mass there, and these are the same number of plants in each pile there. I tell you, this is the kind of data I receive sometimes from the field guys. So it's not always the best. This was a pretty good representation. The corn in the middle, we're looking at the control plant on the left. So everything received the standard chemical treatment. The Sabrex for corn is in the middle. And the K5AS2 on the right is one of our pipeline products, which is bigger still. And these again are shown in the roots on the right here. We have talked a lot about, at ABM we talk a lot about improvements in root biomass, but that's a really hard thing to measure unless you have a rhizotron. So we made up our own rhizotron, and that is a 7-foot PVC pipe that we put into the ground and we fill with soil. And so that's what we did with these, and we treated them with our treatments, the standard chemical treatment plus the treatments that are listed up there on the top, and let them grow through the season, and then pulled those tubes out of the ground and washed away the soil. And so what is shown here is the control, Sabrex, and then 2 pipeline products with a much bigger root mass. And so K5AS2, the pipeline product, and then Omega, which interestingly contains no living biological, contains only a metabolite or signaling molecule from Trichoderma that is formulated here. So this is really interesting because, again, we harvested this at the end of the season, and so what we got here was a season-long effect. That's something we're still working out, how that's happening. So anyway, so these are some of the growth promotion data, and they're just examples. This wasn't quantitative data. I just want to give you the idea because you're probably not used to looking at these kinds of things. Okay, let's move on now, because I've been talking about our products, which our products and our pipeline products, which are 2 strains, sometimes more, but usually the sweet spot is 2 to 4 organisms in there. So I told you I'd talk about the phytobiome. So why do we care about the phytobiome? Well, there are a lot of reasons why we might care about this. First of all, I'm telling you that we're putting a biological product, we're putting biological organisms on the seed into the soil, okay? So, so we know that a corn plant, for example, it's got endophytes living in it. It's gonna have organisms on the leaf tissue as well, right? Some of these are causing disease, some are not. The soil is teeming with life, and we're putting on another living organism onto the seed. Why wouldn't we care about the phytobiome, right? This is actually what we're doing and what we're leveraging in our products. It's just we didn't know it till about 5 years ago because it didn't have a name, okay? But one of the things that we think about all the time is what are we recruiting to the seed when we put our biological there, okay? And so this is all in the realm of phytobiome. And I just want to give a plug here that APS, our professional society, is very heavily involved in phytobiomes. And if you want to learn more about it, if you're not fully into that, then you can learn more here, find the roadmap that's been put together. So, okay. You've probably seen slides like this before. I just went to Web of Science and looked how many times has the term microbiome or phytobiome been used over time. And so there's been a real, a massive increase actually since 2008, 2009. And that's not to say people weren't thinking about this before then, but I think what's happened is that sequencing and informatic techniques have really come along. Like we can acquire so much sequence now. We can, back in the day, people, and sorry I said back in the day, but a long time ago, people were doing PCR on dirt, and they're like, why are you doing PCR on dirt? What are you going to get out of that? Well, this is what we can get out of that, right? We can now look at this with Illumina sequence or some other kind of sequencing technologies, and we can actually figure out a lot about what's there. So there's been a real boom here, and a lot of this has been in medicine, but more and more is in the plant world, and so this is something that's changing. Along with this, along with the scientific discoveries and the the sequencing and bioinformatic technologies that have been on the rise, there's been an explosion in ag biological companies. ABM has been working in this area since 2000, right? And these are the companies, and I know I'm sure I'm missing a lot, actually. These are just the ones I could think of the other day. So I already told you about the big players in the field, Monsanto and Bayer now one, Pioneer, Dow, DuPont, Dow are now one. There's also, so those are the ones who have been around for a while and are traditional chemical companies and plant breeding companies that are now in biologicals. The ones in blue are also companies that are a little bit older. They're kind of on the fringes, kind of second-tier chemical companies or breeding companies that are also playing in this field. And then all of those that are in green, and these are the ones that I know that there are many more of, are the companies that have been started or started working in biologicals in the last 5 years, specifically to leverage phytobiome and microbiome data. Okay, so that's all new. So again, this is showing you kind of the market or the space that ABM is operating in. So every one of these companies is trying to address something that's the fundamental problem in agriculture, that agriculture suffers from the effects of environmental variability, right? There's no question about this. That's why we do breeding. That's why we do trialing all over the country in a lot of different environments. We're trying to address this problem, because if we could get 300 bushel corn every single year, everywhere we planted, well, that's done and I'd be out of a job, okay? But that's not how it is. There's a lot of environmental variability, and this is a plot of some of our data, I think just from last year, showing yields on the y-axis and location on the right. There are probably about 5 corn hybrids that are in this, so it's not all one genotype of the host. But it's showing you that yields vary by location. And there's no question about that. And what's in blue here is our Sabrex per corn product, again, a Trichoderma product. It's increased over the control, or under the control, as the case may be, for down there on the left down here. But what this shows very clearly is that there's no relationship between the performance of a biological and the yield of a corn plant, or even the location. And so why is that? You know, this is a question. Why is that? I don't have the answer, right? And so it's one of the things that we are working on trying to solve, and so is everyone else that I showed on that last slide. Everyone is working on this. So, okay, getting back to phytobiomes, can some of this field level variability be explained by its interaction with the phytobiome? There are a lot of factors out there, but we need to think about what the factors are that we can control and understand, okay? Yeah, ag biologicals interact with rhizosphere and soil inhabitants, the plant endosphere, and the phytosphere. Like this is where that corn plant has to live, or whatever crop that you work on or species. That's what it has to live with. And this is an equation that everyone in here should know. And if you don't, then you should come to love this, because you know you can get a lot of mileage out of this, right? So phenotype, right? That's disease resistance. That's yield. That's stress tolerance. Genotype we think about as being the plant, right? The host crop. Environment, where you plant it, what chemicals did you apply to it? You know, how much rain did you get that year? And then the other part, the G by E. How do the genetics of that host interact with the environment? Because that can, if this term is really big, then you need to go back to doing some breeding and rethinking this, because this is the part that we really want to control, is that genetic by environmental interaction. Okay, so this is the rest of the slide, because what I want to talk about is what can we control here and what can't we control. So there's phenotype, right? I already talked about that. That's the trait. That's what we're interested in the plant looking like at the end of the season. Genotype we've thought of as being the host. Well, there's another genotype here that people are starting to add, and that's the biological product, the microbial, right? And so Monsanto, you know, Pioneer, all these companies, ABM, all of us who are developing microbes, we all have the genome sequences of our strains. We know what the genotypes are. And so what a lot of research is aimed at is how does that microbe interact with the host plant? Where the phytobiome research comes into is how does this interaction, relate to this interaction, right? Because all of this on the end here, that's the environment. I already talked about weather. There's no controlling the weather. Management practices, you may think you control management practices, but when you send your material out to a contract researcher and you say, I would like you to apply 200 pounds of nitrogen, and they apply 3 and don't tell you about it, well, that's something that, you know, it's hard to control. And so you can make recommendations, but in the end, the farmer is going to do what the farmer is going to do because they know their own farm the best. Soil type and environment, can't control that. Rhizosphere biology, what's in the bulk soil, what's in that rhizosphere, right? And I put this with this kind of green and yellow dot here is because I think where we could be going with this phytobiome research is gaining some control or at least understanding of what is in the rhizosphere, right? The rhizosphere is going to be different from what's in the bulk soil. And so if you put a biological on the seed and it's recruiting something or having an antibiotic effect or something, it's changing what would otherwise be in that rhizosphere, right? That's the idea. Can we make this a predictable relationship? Right, that's where all this is going, and that's what everyone is doing who's playing in this field. So, all right, then moving on about how we do the, and this is the part where my talk doesn't really follow the outline that I put up in the beginning. So how we develop strains. We're a small company. There are about 40 employees in ABM, so we need to use our resources wisely. This is a correlation matrix among a lot of different traits that I've assayed for in the greenhouse or in the lab or in field trials with a large set of our strain collection, and that's Trichoderma and Bacillus, okay? So what we want to see is if I can develop assays that are essentially markers for good performance. Okay, I don't want to have to do every single one of these tests every single time I develop a trait. I want to be able to do a few of these assays and have that translate to the field. So that's how we do our well, the beginnings of our strain selection. Then once everything gets out into the field for field trials, well, all bets can be off sometimes. So anyway, the point is there are a lot of different phenotypes that go into a good strain. And this varies at a strain level, not at a species level. I'm sure that you know, you all are aware of that. And this correlation of phenotypic effects is what's gonna get us our high influence strains, and those are the ones that we advance through our process. So that's just to give you an idea of what we're looking at. We're looking at traits. We're trying to streamline how we can select for traits, and then we advance things to the field. So this is an example because we have field trials every year, right? And in 2016, you know, in 2015 is when the phytobiomes groups started really getting going. And so we thought in 2016 in our field trials, this is what we're going to do. We're going to do some sampling of our rhizosphere, and we had actually planned to do some phylosphere as well, but well, ran out of time and resources. But we set up a really nice trial in the field in 2016. We applied seed treatments in the spring when we planted this, and this is silage corn. So we had 4 different biological treatments, 6 corn hybrids, and 4 nitrogen levels because we're testing environmental variability management practices, things like this. And we had 3 reps there, which is fairly typical in a lot of field trials, especially of this size. So, and we had a randomized complete block design. These are our yield results, and again, this is silage, so it's tons per acre. And experimental 1, there's actually that K5AS2 that I had shown previously, and we had significant differences among our treatments there in the field. And just one note is kind of owing to the variability that we see in the field. Often when we analyze data, we use an alpha 0.1. Just so you know, that's how this was analyzed, alpha 0.1, not 0.05, which would be more typical of a lab kind of experiment. So we did this, and I'm going to go a little bit faster. Gosh, I thought I'd get you guys out early. So then we implemented a process, which is to study the rhizosphere of this. And so we collected samples, root rhizosphere samples in the field, isolated DNA, so we could look at 16S and ITS, right, because we're working with fungal products. So we want to make sure that we assay the fungi that are present. We are leveraging the Oxford Nanopore technology, the MinION, because this is a technology that is a good price point. We can bring it in-house and we don't have to wait in line to get our sequencing done wherever we are. And if we decided we want to put something on the sequencer that day, it takes an hour and a half to make the library and we can get it on. So, and that time is coming down to 10 minutes, FYI. So, and then we do the sequencing, and then there's all the downstream, which is the sequence analysis, which I think I can skip over at this point. So, just a brief word about the MinION sequencing. This is if you're not familiar with it, this is a really neat technology that I guess Peter Schweitzer is going to get at the core facility here. You can have reads, like the power users have reported reads up to 2 megabases long for a single read, compared to Illumina, which is 1 to 300, you know, depending on what you're doing here. But the error rates are much higher than Illumina. And so that's a trade-off. But if you're thinking about sequence length versus depth, then that's something that you can think about. This technology is fieldable. If we wanted to, which we don't, we could take this to the field actually and run it off the laptop in the back of the truck. And they're continually improving this. This error rate has actually dropped quite a bit over the last year just because of technological changes with that instrument. So that's a really nice technology. But it also leads to some challenges for us because we're getting far fewer reads than you would for Illumina, and we have a higher error rate. We do get longer reads than Illumina, which means we can look at longer sequences in terms of ITS. We can look at the whole ITS region if we want to. And the other negative we have is that we're interested in looking at fungal communities. And why that's important is because a lot of the software development for analyzing some of these things is for 16S, is for bacterial genomes, not fungal. So. And the other negative is I'm our bioinformaticist, and you know, I'm kind of having to come up to speed in some of this stuff. So looking at CHIME, there's a really clear, okay, for those of you who are not working in this area, CHIME is a software, and actually now there's CHIME 2, which allows you to look at these kind of metagenomic, you know, microbiome type data, and ultimately find where the differences are, or identify what is in your DNA sample, and then find out a little bit about either, you know, well, it doesn't tell you function, but you can really interrogate what some of those different organisms are. You know, this is fine to use if you're working on 16S, and if you have really high read numbers, like with Illumina. But if you're working with MinION data, as we are, sadly, the lower read number and the higher error rate means that CHIME kicks out most of our reads, and we're unable to make it all the way through the pipeline with more than about 9 features. So, so our pipeline is we generate the data, and we use the Oxford Nanopore technology software to do the base calling and quality filtering. And then a BLAST plus analysis and script-based manipulation of the BLAST output, which, in all honesty, is where we need to be, because having the CHIME analysis, looking at reams and reams of data for 16S, isn't necessarily going to get me closer to developing a new product. What I want to understand is a little bit higher level than that, where I don't need to know all the different strains that are there. What I need to actually do in the end is find what the function of that rhizosphere is. And there are definitely tools out there to do that, but in terms of being able to go this far as we are, this is actually what I need to be doing right now. So just to quickly run through massive amounts of data, this is what we found. So we sampled that cornfield in 2016, and we determined a core microbiome by filtering based on bit score. Remember, I was looking at BLAST output. and co-occurrence in all 3 reps. And so we had a lot of data loss, and what I'm showing you here is a 16S results. So we ended up with the core microbiome that was fairly small, and oh, sorry, C is the control, O is omega. Remember that contains no living biological, it's just the metabolite. K is K5AS2, and S is our Sabrex for corn product. So those are the 4 different treatments. And this was a result. So we can see here, so each line in this graph represents an organism, and you're not expected to read the y-axis because most of those are uncultured. And there were just too many of them. And so what's shown here in blue is each line represents one organism. And the color of that line tells me if that organism is represented in one of the treatments or not. So for example, these bars down at the bottom have the control, Sabrex, K5AS2, and omega in them, okay? All 4 colors are represented. These organisms are found in all of the samples. What's interesting is that these are organisms that are found only in the control, and then these organisms up here are found only in either the Sabrex, omega, or K5AS2, or some combination of our treatments. So that was really interesting, right? So that's 16S. And then here's a similar, although less organized representation of what we found in, sorry, did I say 16S? That was 16S, this is ITS, okay. Similar representation of what we found for the ITS community. Again, there were several genera, so this is on a genus level that these are listed here, that were present in all treatments. Some that were in most of the treatments, but there are some that are present only in the control, only in Sabrex, only in K5AS2, and only in omega. So what this is telling us is that we saw changes in the rhizosphere community that were correlated with treatments. And this was a replicated study. And so I feel pretty good about this because really we just wanted to see, are we moving that rhizosphere community away from what it is as a control? Do we have the ability to change it? Because it gets to mechanism. What are our organisms doing? Do they only have an effect on plant? Because I told you at the outset that Trichoderma changes plant gene expression. Is that all they're doing? Well, not necessarily, right? They are producing metabolites. That top bar up there is omega. This is just a metabolite produced by Trichoderma that we've applied to the seed, okay? So are our organisms communicating with the other microbes in the soil? Probably so. Is it changing how the plant communicates with the organisms in the soil? Maybe so, right? What this does is get a lot more questions, really. So this is another way of looking at the ITS data, and I just did this yesterday because, you know, as I said, the thing we really want to do is look at the change in function of the rhizosphere, right? Are we changing anything functionally here? And so I just classified the output from the ITS analysis into different kind of lifestyles or functions there in the rhizosphere. And so those are very, very general. And what you can see is, and it's based on the genus level. So for example, that first blue category there, beneficial, pathogen, that means in that genus, we find both beneficial and pathogenic organisms. It's a real mix, and you can't really say, oh, it's mostly this or that. So you can see in the control, mostly a mix, but you know, there's what's notable about this is that there's more diversity in the control treatment than in the Sabrex, omega, or K5AS2. Which is interesting because we think about ag chemistry as being something that reduces diversity in our crops, reduces soil diversity, reduces the, you know, foliar phyllosphere community, reduces that diversity, and we think of that as being potentially bad. yes, we want to get rid of the pathogens, but we don't want to get rid of everything else. And so seeing a reduction in diversity here was kind of interesting to me because, okay, that, you know, this is not that it's the same thing as what chemicals do, but it's still a reduction in diversity. The other fascinating thing here is a real increase in what I called pathogen, right? And again, I called things pathogen based on their genus, right? That said, there are a lot of things like, for example, in Fusarium, there are pathogens there, and there are things that are not pathogenic. So what we've really done is just change this distribution, but it definitely argues against kind of a niche replacement model here. So this analysis, as with all the other analyses that I've just shown you, begets more questions than it does answers. But it really makes us start thinking about what our products and what a biological might contribute to ag. So with that, I would like to turn over if there's any time for questions. But you know, my summary is pretty simple. I mean, you know, we're assembling strain consortia that have an effect and a beneficial effect on crop production. I think what's going to be really valuable going forward is assembling very large phenotypic data sets. I think probably everyone in this room here knows that that's where the bottleneck is in making progress with all the sequence data, is we need to have the phenotypic data that goes right along with it. And then this is my teeny tiny little conclusion from what we did in the 2016 trial, is that our seed treatments that we used in that corn trial appear to influence the Rhizosphere community membership. And that's about all I can say. So with that, if there's any time for questions, I'd be happy to take some.
Questions here at Ithaca? I was wondering if you've done any studies on what happens long term if you're treating a field with something over and over and over, even if it's just on the seed.
You mean a biological product? Yeah, so that's something that we're initiating now, but I know that Dr. Harmon has done that where applying something on a winter rye, so planting it in the fall and then planting back in the spring. I think he did this with raspberries, and showing that where the rye had been inoculated in the fall, then they had more Trichoderma on the raspberries when they planted them that spring. That said, and we also have lots of data on winter wheat, which is planted in the fall and then you see it in the spring again. But typically, when we think about perennial crops or something like this, our recommendation is to treat every year. Well, yeah, we're a company, we want to sell it, right? But those are data that we are still in the process of acquiring. So, yeah, Phil.
Paul, you mentioned that there was an effect of Trichoderma on gene expression in the plant. I have two questions. One is, what is the magnitude of that change in gene expression? And then, do you see a similar type of gene expression done in different environments with that particular microbe?
Okay, to the environment question first. No, we haven't tested it in a lot of different environments, but a lot of different people around the world have, and it's mainly greenhouse environment is where that's being tested. And so it is something that's reproducible, seen over and over again. Magnitude, they are significant, and I'm just trying to, you know, I mean, statistically significant. And I'm afraid I don't have the specifics on that, but
Tens, hundreds, thousands.
Oh, tens. Yeah. Wait, number of genes or magnitude of change?
Number of genes.
Oh, hundreds. Yeah.
Okay.
Yeah. Mm-hmm. Yeah.
Polly, I appreciate that you have a very challenging problem. It seems, if I understand you correctly, that the phytobiome varies from field to field, just to start with. And what you're trying to do is to change it to make it more favorable for plant growth. Mm-hmm. And so you try to match the organisms that are contributing to the particular field. And once you do that, in 1 year, you change the phytobiome, and presumably if you keep doing that year after year, then you have the perfect phytobiome, and you're out of business for that particular preparation. Is that right?
Well, yes and no. I would say that, okay. So first of all, this is something that's in its infancy. Can we actually shift the microbial community in a given field over time such that it becomes a generally agriculturally conducive field? Well, that remains to be seen, and I think that there should be a lot more work on organic soils and things like this, but have all the microbial communities been characterized there. And it's a problem of environments, right? Because every field is different. And so how do we assay every single one of these fields to determine to get the ability to make a global shift, to say that, okay, what you need to do in this field is apply this product year over year over year, and then move this into something that's a preferred microbial community, and so then it's going to be the perfect field. So am I worried about putting us out of business because if people start using our products, then they're just going to improve their soils and that'll be that? Not really. because that's a long way off, and that's a long way off before we can say all that. The other thing that happens is weather, right? You have a hot, dry year, even though you've done all this work to try and improve your soils, you could kill it right there, right? You could change everything in a single year. So, or you have a disease come through, or an insect or something like this that brings another microbe, changes the chemistry of the plant that's actually in that soil. Remember, you're planting plants in here too, and that has influence as well. So I'm not worried about my job security in that regard. There are other reasons why I might worry about that. But I think that figuring all this out is gonna take some time. I may be wrong, but because it's a systems biology problem that also has weather, right? It's not just biology, it's weather, it's soil environment, it's everything. So there are human factors as well. So once we let all the data scientists just be in charge of everything, then we'll probably be fine. But we're a long way from that.
Can I ask if there are any questions in Geneva or other locations? Okay. All right. Thank you, Molly.
This has been a production of Cornell University, on the web at cornell.edu.